So as you have seen in my thumbnail, today I'll be talking about Wobble hypothesis. It's a very important central dogma process, or you cannot say central dogma process, but it's a mechanism in central dogma process, which is giving the flexibility to the cell to go beyond its capacity. So let's start. What is Wobble hypothesis? So Wobble hypothesis or Wobble pairing is a pairing which defies or which is separate from the Watson-Crick pairing. So this pairing, we know that A bonds with T, G bonds with C, or in case of RNA, it is U. So it is a normal process of bonding that you already know. But in case of Wobble hypothesis or Wobble pairing, there is a flexibility of the bases which can pair with. So there will be, you will be seeing the difference in their pairing ability with beyond the normal pairing. That is Wobble hypothesis, or that above is the Wobble pairing. So I'll give you some examples and draw you diagrams so that it will be easy for you. So here, one more base pair I'm going to introduce, that is inosinate. I is equal to inosinate. So inosinate is a nucleotide which contains hypoxanthine as a base. So inosinate contains hypoxanthine as a base. Now, Wobble hypothesis or Wobble pairing, if you see, this is RNA 5' to 3', and suppose this is the tRNA, that is 3' to 5'. Now, the first two bases, Wobble research says that the first two bases of the mRNA, that is the mRNA, and this is tRNA. So corresponding codon and anticodon. So you know that the anticodon is present in the tRNA. So here I'm going to introduce one pair, that is AGC. Suppose it is present in the mRNA, so it is a codon. Now against this, there will be an anticodon, that is GCU, as it is a RNA. Now, if you take this as a 1, 2, 3, and here it will be 1, 2, 3, the number of sequential number of the codons. So the first two of the codon, the nucleotides present in the mRNA, the first two codon is highly specific. Strong bonding, encourages strong bonding. Whereas the third one is flexible. So same here, in case of 2 and 3, it is highly HS, I'm writing sort to save time, that is highly specific, whereas the A, the number 1, is flexible. So we have read that one amino acid can be coded by 2 or 3 codons, or more than 1 codons. Now specific tRNA is present for each and every amino acid. But if one amino acid is coded by more than 1 codon, that time it creates a more, or it will engage the cell to create more tRNAs, which will now create a chaos or use of extra energy for producing a protein incorporating the particular amino acid, which may be coded by more than 1 codon. So to reduce or minimize this thing, cell or nature compromises this thing as in the form of Wobble pairing, and the tRNA the anticodon present in the tRNA contains the, in case of G, if I replace this with I, now I am replacing the G with I, that is the inosinate present in the anticodon. Now this I can pair with C, G, C, A and U. So this inosinate can pair with CAU. So if you change the codon in mRNA, that is, first two will be the same, but the third one has been changed from C to A. That time it will bond with the I. U also bond with the I. So this gives the flexibility into the cell or to the mechanism of protein synthesis of the RNA-tRNA bonding pattern, so that the one tRNA can recognize a separate codon, or the variation in codon is allowed by this Wobble hypothesis or Wobble pairing. Now I'll give you a clear concept of one codon recognized, two codon recognized, and three codon recognized, how it is functioning with the help of Wobble based pairing. Okay, so one by one. One codon recognized in case of, suppose this is tRNA, now mRNA, 3 prime, 5 prime, so here suppose U, A, G, so here will be A, U, C. This is one case. Next one is the, I'm writing T and A only to save time. 3 prime, 5 prime, U, A, U, A, U, A. So this is one codon recognized. If one codon recognized, then this will be the pairing. So you see the that is the 1, 2, 3 here and 1, 2, 3 here. So this one remains the same as Watson-Crick base pairing. Now, if two codons are recognized, two codons to be recognized, that time T 3 prime, U, C, G, 5 prime, mRNA, 5 prime, here A, G, U and C. So here you can see the difference, that the G base pair cells in case of Wobble pairing, you can also recognize uracil, which is generally paired with adenine, is now recognizing a bonding with G apart from the C. And another one is the tRNA 3 prime, UC, U, 5 prime, N, 5 prime, A, U, A, and also G. So see the difference here, that apart from the Watson-Crick base pairing, that single base is recognized by more than, which is not normal, more than one nucleotide. So here two codons is recognized. Now, if three codons to be recognized, well, one example enough for here, that is CAI. Suppose and 5 mRNA, 5 prime, here G, U, in case of I, separate A, U, C. So see the flexibility of inosinate with these three bases. So inosinate can be base paired with all the three nucleotides. That is the variation or the beauty of Wobble pairing, which gives the flexibility and encourages the single tRNA to recognize more amino acids. So that is Wobble hypothesis. So it's differ from Watson-Crick base pair, but it is highly essential in protein synthesis. So I hope that is clear. If you have any questions, please ask me. So if you like my classes, give a big thumbs up and don't forget to subscribe my channel because these things stimulate me to take futuristic classes for you guys. So thank you and be with me.